Hello, our today's topic is from Fermentation Technology, paper MB335 for topic Endotoxin Detection, chapter Quality Assurance of Fermentation Product. This is your sixth topic of fermentation technology, and in this pyrogen testing, that is endotoxin detection, is included. This one is for the test LAR test. About this LAR testing, this is also called as a limulus amoebocyte lysate testing, and the test is mainly used for detection of endotoxins. This is carried out in the laboratory, that is in vitro assay, and here this one is used to detect presence and concentration of a bacterial endotoxins which are present in various pharmaceutical products, as well as this can be used for certain devices which are used by medical industry, like catheters, as well as for the syringes. This is important part of pharmaceutical microbiology. Now, before studying this LAR test, we'll see some history of it. Frederick Bang and Jack Levin, they observed that blood cells from horseshoe crabs were found to clot in the presence of endotoxin. This one is a picture of this horseshoe crab, and here this one is very unique type of a phenomenon, that is the blood cells of these crabs are able to clot in the presence of endotoxins from bacteria, and this technology was used in the development of this endotoxin detection assays. So based on this, there is a LAR test. LAR test ensures that sterile pharmaceutical products are safe for human use. This is significance of the test. That is, number of a biological products which are prepared by pharmaceutical industries, whether they contain endotoxins or not, are detected in quality assurance laboratories of that particular pharmaceutical industry. So this LAR test is recommended in all international pharmacopoeias as the method for finding bacterial endotoxins. The LAR test was commercially introduced in 1970s, but nowadays also it is used widely to find out pyrogens, endotoxins present in biological products. The reason the bacterial endotoxin test is called as a LAR test is because of Limulus amoebocyte lysate testing, because the lysate from blood cells, that is amoebocytes from horseshoe crabs, they are used for this purpose. The lysate from this horseshoe crab blood cells react with the bacterial endotoxin. They can detect very small quantity of endotoxins which are present in the pharmaceutical products. Endotoxins are nothing but lipopolysaccharides which are present in the cell wall of a gram-negative microorganisms, and they can be detected by this test. These endotoxins are a type of pyrogen, that is the substances which when present in the body, when they released, at that time there is a febrile illness, temperature of body increases. These are nothing but as you know, lipopolysaccharides present in a cell wall of a gram-negative microorganisms. But there are some exceptions like Bacillus thuringiensis. This is gram-positive organism, but then also it produces delta toxin, which is endotoxin, and this can be detected by this LAR test. Endotoxins are structural components of the bacteria, and they are released when there is a lysis of a bacterial cells. So when infection occur in human being by gram-negative organisms, at that time, after some time, there is a autolysis of these gram-negative organisms, and at that time these endotoxins are liberated, and because of which level of cytokines increases, and this is nothing but a appearance of a febrile illness in the patient. So it is necessary to find out whether such a type of endotoxins are present in the given pharmaceutical products, drugs, saline, whatever the biological product which is used in the treatment of a patient. Not only that, in devices like syringes, injectables, they may present, and so it is one essential test. These endotoxins are toxic for human beings as well as for animals, because they are responsible for a pyrogenic response, rise in body temperature. Not only this, okay, if they are present above certain concentration, it become fatal. Fatal, that is, mortality rate is quite high if level of endotoxin increases, as it hamper not less than 30 biological activities of a human being. This leads to death of the cells. This is by initiating complement activation, and so it is necessary to detect them. So endotoxin tests are performed in pharmaceutical and medical device industries, not only for the finished product but also for the raw incoming process material, because possibilities are there whatever raw material which enter in that particular industry, it already may contain endotoxin. At the same time, during production of the final product, finished product, chances of a contamination and growth of gram-negative organism, okay, it will be there, and so endotoxins may present in the last finished product. So here these tests are carried out for raw in-process material, at the same time for the finished product. What is responsible for the this LAL test, positive LAL test? So here coagulation of a hemolymph in this limulus polyphemus crabs are the amoebocytes, which is the main component of the hemolymph of this animal. And here manufacturer separate this factor amoebocyte from it and label it as a LAL reagent. And here in vivo condition, that is when it is present in the body of that horseshoe crab, it is one part of its immune defense mechanism, because they are invertebrates, and in this invertebrates, these amoebocytes fulfill the function of a white blood cells in the vertebrate like us. They defend the organism against pathogens. Therefore, they respond to the influx of endotoxins from the bacteria by releasing a series of enzymes, because of which clotting of it occur, neutralization of it occur. Scientists studied this phenomenon until ascertaining that if a lysate of the amoebocyte extracted from the horseshoe crab in a watery environment is diluted, and this could serve to detect very small quantities of endotoxin. See, organisms are very small. Upon lysis of it, very small quantity of a lipopolysaccharides liberated. But here characteristic of this LAL test is that it can detect very small quantity of endotoxins. Here, these amoebocytes contain procoagulant enzymes. Basis of this reaction is based on these procoagulant enzymes that trigger a chain of reactions. The final product of this chain reaction is a gel comprised by coagulated proteins. The enzymatic response is produced when the amoebocytes enter into contact with the endotoxins. And here the test is now positive, where there is a just coagulation of the endotoxins of the hemolymph by the endotoxins. A negative LAL test only indicate the absence of endotoxins and not the absence of other pyrogens. So this is limitation of the LAL test. That is, only lipopolysaccharides from gram-negative organisms, they are detected by the LAL test, but not other pyrogenic substances which are of some other origin. Now, based on this lab test, a number of versions of a types are identified yet. From that, first one is a gel clot technique, second one is a turbidometric method, third one is a chromogenic method. All these three tests, they are widely used. First one, that is gelation method, gel clot technique. It is mainly for a qualitative, that is to identify whether endotoxins are present or not. While chromogenic method and the turbidometric method, they are qualitative as well as having quantitative approach. In the chromogenic method, there is a development of a color after cleavage of a synthetic peptide chromogen complex, which can be measured photometrically, colorimetrically. While in the turbidometric method, after cleavage of an endogenous substrate, turbidity is formed. Again, can be measured optical density, that is photometrically, presence of endotoxins and their level in the particular pharmaceutical product. Both methods, chromogenic method and the turbidometric method, can be performed in two ways. First one is a endpoint method and second is a kinetic method. Means chromogenic endpoint method and chromogenic kinetic method, and same time turbidometric endpoint method and turbidometric kinetic method, they are possible. Gel clot technique is based on gel formation, that is gelation, and it is very simple method. Positive means endotoxins are present, negative means endotoxins are absent. And this gel clot is now considered as a qualitative and up to certain extent semi-quantitative, but not like chromogenic method and turbidometric method. It able to measure exact quantity of endotoxins present in the sample. The gel clot assay is run in tubes that are placed in a water bath or in dry heated oven at 37 degrees Celsius. Water bath is always preferred one. Only after mixing the sample, pharmaceutical sample, with lal reagent, after 1 hour incubation, the tubes are flipped 180 degrees Celsius, and a firm clot, which is if formed, it stays in the bottom of the tube, indicate positive reaction. So very simple principle of it, that is just clotting of the mixture occur, and that firm clot remain as it is at the bottom of the tube. If the liquid flows when you flipped it, that tube at 180 degree, and if liquid flows down the side of the tube, the result is now called as considered as a negative for presence of endotoxin. So in this way, gel clot method is based on presence or absence of a gel clot in the sample tube. Here, the basis of this test is gelation occurs when proteins are coagulated due to presence of endotoxins. Means whatever the proteins present in the hemolymph of that horseshoe crab, they get coagulated if endotoxins are present. The detection limit of this test depend on the manufacturer who prepared that lal reagent. And using this gel clot method, the detection limit is normally between 0.01 and 0.03 endotoxin units per ml of solution used in the test. This means that solid gel does not come to be formed below this concentration of endotoxin when moving the test tube. Again, this one is limitation, means if it is less than that, the test is negative. A criteria used in this method is to turn the test tube at 180 degrees Celsius and ascertain that the gel remains intact. And so this is as it measures from 0.01 to 0.03 units of the endotoxins, it is up to certain extent semi-quantitative one. Now, as you know, there are two more methods, turbidometric and chromogenic method. We'll see turbidometric first. This technique is a photometric assay, measuring increase in the reactant turbidity. Means more endotoxins, you can see more turbidity in the sample. On the basis of a particular assay principle employed, this technique may be classified as an endpoint turbidometric assay and a kinetic turbidometric assay. Now, here basis of this test is very simple. First, we have to prepare one standard graph using known quantity of endotoxin. Means known quantity of endotoxin plus in that we have to add that lal reagent, and in the serial order find out exact quantity, exact optical density obtained upon addition of that increased level of the endotoxin. Prepare standard graph and then plot whatever OD of the test on it so that we'll get exact quantity of the endotoxin present in the sample. The endpoint turbidometric assay is based on the quantitative relationship between the concentration of endotoxin and the turbidity which is developed. This can be measured in the form of absorption or absorbance and or transmission of the reaction mixture at the end of incubation period. While in the kinetic turbidometric assay, it is a method to measure either the time, here onset time you have to consider, needed to reach a predetermined absorbance or a transmission of the reaction mixture. This is the rate of a turbidity development. This can be measured in this kinetic turbidometric assay. Here incubation temperature is usually 37 plus minus 1 degree Celsius. This one is according to or recommendations by the lysate manufacturer. Now, similar to that, chromogenic techniques can be used, basis of which only difference is if you compare with the turbidometric assay is that in that case we are measuring turbidity, while here change in color or intensity of the color is measured based on the release of chromophores. This is the technique is an assay to measure the chromophore release from suitable chromogenic peptide by the reaction of an endotoxin with lysate. In that case, turbidity is measured. Here, chromophore released, they are measured. On the basis of a particular assay principle employed, this technique again classified as either an endpoint chromogenic assay or a kinetic chromogenic assay, similar to the turbidometric method. The endpoint chromogenic assay is based on the quantitative relationship between the concentration of endotoxin and the release of a chromophore at the end of incubation period. Again, here there is a requirement of a preparation of a standard graph. Known quantity of endotoxins are allowed to mix with the lal reagent and graph is prepared. On that, one can plot results of their test samples and can find out the exact endotoxins present in the sample. The kinetic chromogenic assay is a method to measure either the time, onset time, needed to reach a predetermined absorbance of the reaction mixture or the rate of color development. So in this way, the lal test can be performed by three-way gel clot method, chromogenic method, and turbidity method. We can summarize this. The lal, that is limulus amoebocyte lysate testing, is also known as a bacterial endotoxin testing. This lal term is derived from the blood cells or amoebocytes of the horseshoe crab Limulus polyphemus. The lysate from horseshoe crab's blood cells react with the bacterial endotoxin. Bacterial endotoxins are nothing but lipopolysaccharides of a gram-negative organisms, and exceptionally endotoxins, delta toxins from a Bacillus thuringiensis like gram-positive microorganism. all can be detected by using this LAR test. At the end, there are total 3 methods of LAR test: gel clot LAR test or regulation method. Second one is a turbidometric method, and third one is a chromogenic technique. First one, that is gel clot method, is a qualitative or a semi-quantitative simple method, and the turbidometric method and the chromogenic method, these are qualitative as well as a quantitative method. And here positive control and negative control are required during this testing. And here preparation of a standard graph is a main part of a turbidometric and a chromogenic technique. So based on that, one can find out exact quantity of a endotoxin present in the pharmaceutical product, which is important test in the QA department of any pharmaceutical industry. So this is all about LAR test. Thank you very much.